Good afternoon, all. Yesterday we had discussed about the uh, immunoglobulin, Y-shaped molecule, and antigen binding site, okay, in the V shape, as well as we had discussed with the epitope and paratop. Let us discuss with the one point, as you know that in the biomolecules, the structural component of the protein is amino acid, and in amino acid one side is known as N terminal because ammonium ion is there or amino group is there, and on opposite side carboxyl acid group is there. Fine. So here in antibody, in Y-shaped molecule, here on the antibody structure in the Y-shaped molecule, this portion is known as N terminal because at this region NH3 plus is there. Okay, whereas here at this bottom side COO minus is there. So this is known as C terminal. Okay, fine. The immune system based on the B lymphocytes. You know that B lymphocytes produce antibody and responsible for HIR or AMI. So here the proportion is 10 to 20 percentage. So here 10 to 20 percentage proportion, that is consists of the B lymphocytes in the immune system. Now functions of antibody. Functions of antibody in that Here, functions of antibody. First function, that is known as agglutination. You know that antigen is known as agglutinogen, antibody is known as agglutinin. So antibody joined with antigen, and antigen antibody reaction occur, that is known as agglutination. Second function, opsonization. So agglutination, in that you only remember that thing, cell lysis. Fine, by attachment of the antigen with antibody. Second is opsonization. Opsonin antibody is secreted. Opsonin antibodies are secreted IgG and IgM, and with the help of this antibody, antigens are killed. And third is neutralization. Toxic substances. Toxic substances secreted by virus or bacteria can be neutralized, fine, with the help of this. Now, tell you the functions of the different antibodies. You know that 5 types of antibodies are there. Short form we say that Gamma, IgG, protects body fluid, IgA protects body surface, IgM protects body bloodstream, IgD activates B lymphocytes, and IgE mediates regional hypersensitivity. Fine. So these are the basic functions of the gamma, IgG, IgA, IgM, IgE and IgE. Skin and mucous membrane are first line defense. First of all, the antigen which is present first time to defense mechanism, that is skin and mucous membrane. So skin and mucous membrane are first line defense. Neutrophils, monocytes, macrophages, interferons, and fever. These are second line defense. If the antigen is coming in contact with the first line and pass through it, then coming in contact with the second line effect, and specific immunity by specific immunity by B and T lymphocytes. Fine. That is HIR, AMI or CMI. Fine. These all are included in third line defense. Now, let us discuss with the specific antibody in detail. Few points. IgG, only MCQ related points I can tell you. Fine. Most abundant amount of the antibody, highest proportion, that is 75 to 80%, 75 to 80% major proportion of all the antibodies, fine, that is IgG, and it has ability or capacity to pass through the placenta. So IgG pass through the placenta, reach to the embryo and give the protection to the embryo against particular infection, as well as in adult person it promotes phagocytosis process. Fine. In this way it is helpful for immunity power. IgA. IgA antibody present in 10% only. It is primary antibody present in the colostrum. As you know that colostrum is the first milk secreted from the mammary gland after delivery of the child, and apart from the colostrum, you know that colostrum is the richest source of the IgA antibody. Fine. And apart from the colostrum, IgA is present in saliva, sweat, mucus and tears. Fine, etc. IgM. IgM is the oldest and first antibody generated against the antigen. If any type of the antigen present enter inside the body, first of all IgM antibody is secreted. is largest sized Ig immunoglobulin, pentametric in form and highest molecular weight. Or heaviest molecular weight. Now, IgD. Present in trace amount. Less amount 1 to 3% only, and as you know that IgD activates the B lymphocytes and produce the antibody. So only this one thing for IgD. Now IgE. Present in very small quantity, 0.05% only, a very less amount of IgE. It has highest ability, strong ability to bind with mast cells and basophils. As you know that during allergic reaction, IgE antibody is secreted, and from the mast cells, heparin, histamine and serotonin. If you have remember in 11th standard, areolar tissue, majority for any type of the allergic reaction, mast cells secrete histamine and serotonin, and basophil, generally basophils are absent in healthy individual, but in allergic reaction basophils are located there. Fine. So basophils consist the S-shaped nucleus. So these are about the detailed information of the different antibodies, and according to that different MCQs related thing. Natural killer cell, short form NK cells. Natural killer cell is non-phagocytic, large granular lymphocyte. Fine. So this is about the detailed information of the antibody and related MCQs. Generally, to improve the immunity power, we use the vaccines. Fine. So in vaccine, in vaccination and immunization, attenuated or inactive pathogen are, we say that inactivated or heat-killed pathogen or antigenic protein of the pathogen in a limited quantity. These are used inside this. Edward Jenner, okay, Edward Jenner, known as father of immunity. Edward Jenner, known as father of immunity, first time he discovered smallpox vaccine known as Citra, right? Then Louis Pasteur, Louis Pasteur discovered the process of inactivating the pathogen, as well as he prepared the vaccine for chicken cholera, anthrax, and rabies. Von Behring mainly focus on passive immunization and prepared anti-diphtheria serum ADS. That's why passive immunization is promoted by von Behring. That's why he is known as father of passive immunization. Hepatitis B vaccine. Hepatitis B vaccine discovered by experiment on the transgenic yeast, and here the transgenic yeast can be formed by the biotechnology or genetic engineering method, or we say that recombinant DNA technology, in short form small r DNA technology. Okay. Okay, now discuss about the different types of vaccination. I can form the table for this. Here, the types of vaccine, it consists these types: live vaccine or natural or attenuated vaccine, you say like that. Killed vaccine, that is inactivated one. Toxoid vaccines, in that we introduce the toxic substances. And recombinant antigen vaccine, known as subunit vaccine. Here, live vaccine in the examples are smallpox, rotavirus, BCG vaccine, Bacillus calmette-guerin vaccine. But I can move on one side here. BCG vaccine is mostly used for TB patients. Most important NEET MCQ. BCG vaccine is used for TB patients. TB, full form tuberculosis. Generally, tuberculosis is very common disease of the lungs, and tuberculosis mostly caused by the Mycobacterium tuberculin named bacteria. So BCG stands for Bacillus calmette-guerin. Actually, here we say that trivalent vaccine. In Gujarati we say that Triguni Rashi. Fine. So here generally nowadays pentavalent vaccine is also there. In that vaccine, the combination of the 5 different diseases vaccination is mixed there. Fine. So BCG is live vaccine, then OPV. Oral polio vaccine. Okay. In oral polio vaccine, 2 drops of the vaccine kept in the mouth. Fine. MMR. Measles, mumps, and rubella. measles, ori, mumps, dal kachori kehવાય. And as you know that the mumps caused by infection to the parotid gland. As you know that the parotid gland is the largest salivary gland present here nearer to the ear. Fine. So if any type of the infection to the parotid gland, the largest salivary gland, leads to mumps. Fine. So these all are the live vaccine. Now Salk polio vaccine. IPV stands for injected polio vaccine. Here, inactivated poliovirus can be injected in the proper dose. Fine. Whooping cough, pertussis, whooping cough, pertussis known as kali khasi, typhoid, rabies, influenza, pneumonia, and cholera. These all are killed vaccine or inactivated vaccines. Toxoid vaccine, tetanus, TT vaccine, short form, diphtheria, we say that DT vaccine. Okay. Generally, diphtheria vaccine, it is toxoid one. Actually, DPT vaccine is there. Diphtheria, pertussis, and tetanus, and botulism. Okay, generally DPT vaccine is there. Diphtheria, pertussis, and tetanus vaccine. Fine. So here in that diphtheria and tetanus, we use the toxoid substances, whereas heat killed whooping cough, fine, pathogens are introduced at DPT vaccine, and recombinant antigen vaccine, that is hepatitis B. So this is the very important types of vaccine table for you. Okay, so here these are the different types of the vaccine. So I hope you understand this one. Remaining topic we will discuss in the next lecture. Thank you very much to all to being with me and understand these topics for NEET. Thank you very much to all. See you in next lecture.